Welcome students. I hope you all are doing well. Welcome to MDCAT Biology 2025 lectures, where I'm going to share lectures of different topics with you. We're going to start with biology, but definitely we're going to cover physics and chemistry too. All right, so let me just share my screen. Yeah. Okay. So basically, we have to start from the first topic that is given to us, and the name of the topic is acellular life, right? And there are basically two components which we have to cover over here. The first one is viruses. Okay, now what do we have to cover in viruses? We have to see their classify them on the basis of their structure, strands, diseases, and hosts. Okay, you only have to do the classification for the MDCAT. You don't have to do anything else, but we're going to see the definition and a bit of structure and a background of virus, okay, so that you can recall that from your FSC knowledge. So let us start with the overview and structure. What is a virus? I'm going to ask this question to you. Virus is basically it is basically a microscopic agent that replicates only inside the living cell. Okay, this word only is very important. Why? Because virus is obligate intracellular parasite, right? It means that there are some parasites which are obligate and some of them are facultative, right? So facultative means that it can live with or without. There is an option. But whenever we say obligate, it means that it definitely needs this thing in order to survive. It is definitely based on that condition, right? So obligate intracellular means that it cannot live without the presence of a host, right? If there is no host present, so viruses will not be able to survive. And what is a virion? This can come in your MCQs. It is a mature infectious virus particle. When the virus has, infected the host and it has become mature inside the host cell body, then it is known as a virion. Right? Now, what is the structure of a virion? I want you to have a look at this diagram. Right? First, what is present on the inside? Inside, we have the nucleic acid or the genetic code, that is DNA and the RNA. Right? There is a central nucleic acid core which is present, and this is known as the genome, right? It does not have a proper fixed nucleus the way other cells have, right? Okay, and after that it is surrounded by a protein coat, and then this protein coat is made up of capsomeres. See, you see this yellow coat? This is the protein coat, right? This is known as what? This is known as the capsid. And capsid is made up of units, simple units that are known as the capsomeres. Okay. These are the protein subunits as you can see over here. Okay. Now we move on to the examples of capsomeres. You don't have to learn these, but just keep them in your mind that herpes virus is made up of 162 capsomeres. It means that the capsid which surrounds the DNA or the RNA genome, it is made up of 162 basic simple units. Similarly, for adenovirus, we have got 252 capsomeres, right? So you're getting me. Next, we move on to the nucleocapsid. What is nucleocapsid? It means nucleic acid along with the capsid. It is known as the nucleocapsid. See, after this capsid, we have got the envelope, right? Now, what is this envelope made up of? This envelope is made up of lipoproteins, okay? And why is this made? So that it can offer protection against the host cell, right? And it is used for easy survival inside the host cell body. Okay, it is covered with what? It is covered with glycoprotein spikes to help it recognize the host cell. See, these are the spikes which you can see, and they cover this envelope, right? And in this way, they can attach to cell surfaces. Okay, these are just like a hook. Okay, and they help to attach to different surfaces so that they can bind to the host cell and then invade it. Right now, there are some viruses which don't have envelope. This layer, the green layer, is absent in those viruses, and those are known as naked viruses. Okay, when a person doesn't wear clothes, he is known as a naked. Similarly, when a virus, it does not have envelope, it is known as a naked virus. Right? Next, we move on to the capsid symmetry in virus. Okay, so now what is a capsid? As we discussed earlier, that it is the protein coat made up of capsomeres, and there are different kinds of symmetry of this protein coat in which these are arranged. The first one is isosahedral. Wait, I'm going to show this to you. wait a second. See, this green one, as you can see, this is known as the icosahedral, right? Now let us see. The cap is shaped like an icosahedron. What does it mean? It means 20 triangular faces and 12 vertices. Right, and it encloses nucleic acid in a small volume. It's just like, this, a pyramid, a diamond, whatever structure you can say. And this is a very small surface area in which it can enclose the nucleic acid material. And these are the examples which you need to remember. Why? Because viruses are classified on the basis of the symmetry of capsid, right? So on the basis of symmetry of capsid, that have a shape of an icosahedron, these are adenovirus, herpes virus, and poliovirus. So you need to remember these examples. You need to remember what is icosahedron, that it has 20 triangular faces and it has got how many vertices? 12 vertices, right? I hope you are clear between the concept of faces and vertices. If not, please type in the comment section so that I can explain that. The next one is the helical symmetry. Now let's move on to the helix. Yep. This is known as the helical symmetry. Also, when we were studying DNA and RNA, we studied that DNA has a helical structure, right? So helix means this kind of structure. See, this structure is known as a helix, right? This is the capsid which has to enclose this helical virus and this, wavy kind of DNA. And it is made up of units that are known as the capsomere. So icosahedron has a shape like this, 20 faces and 12 vertices. Helical virus, it is just like a cylinder. Now let us read. These are arranged in a spiral around the nucleic acid. They form a rod-shaped structure or just like a filament, right? This is a rod-shaped structure which the helical viruses form. Now you have to remember the examples. The classic example of this is the TMV, which is the tobacco mosaic virus, right? That infects plants. Next we have rabies and influenza. So you have to remember these are very important for your MCQ point of view. Next, we move on to complex symmetry. Now, what is complex symmetry? It does not fit into any particular shape. It has an irregular shape which is very complicated as compared to other viruses, right? So, it is often present in a combination. For example, the head is of icosahedral symmetry, just like this, and then tail, it has got a helical symmetry, right? Example is bacteriophage, that is TEV phages or pox virus. Now, what is the structure of a bacteriophage? I'm going to show this to you. See, this is the head of the capsid. You see the similarity between this icosahedron and this, the head of the bacteriophage? Both of them have a similar shape. Both of them are made in this kind of structure, and they have 20 faces and 12 vertices. But you see that this sheath and this tail, it has a structure of a helical virus. See the similarity. So this is what? This is a complex, this is a virus with a complex capsid symmetry. Okay, complex structure. Why? Because it is a combination of two or more structures. Right? Now we understand the bacteriophage anatomy, that it contains the capsid head, the collar, the sheath, the base plate. This is the base plate, and then we have the tail fibers. Sorry, earlier I said that this is the tail. No, not the tail. This is known as the sheath. So the shape of the sheath is similar to that of the helical virus. That is why it is known as complex symmetry. Why? Because it contains more than one structure. Okay. Next, we move on to the classification of virus. How are viruses classified? First and foremost, we need to see the Baltimore classification. You need to remember this name, that how are viruses classified on the basis of DNA and RNA, and that too double-stranded DNA or single-stranded. Okay. See, you have to remember all these 7 groups. These are basically, you just have to remember them. This is rote learning, okay? It's not much of explanation. And you have to remember the example specifically, that double-stranded DNA is present in smallpox, double-stranded DNA is present in herpes virus, okay? So you just have to learn them. Please take a screenshot of these. Some of the examples are very common. For example, this coronavirus, it is a single-stranded RNA virus, right? And also this one, hepatitis B, HIV, which we are going to study later, and This is also a part of your MDCAT syllabus. So please remember this, that HIV is a part of positive single-stranded RNA virus. Okay, please remember the common examples, right? So the first class, as you can see, there are 7 classes of the Baltimore classification, and the first class is dsDNA. Okay, double-stranded DNA. Second class, single-stranded DNA. First, we are going to take the DNA. Double-stranded DNA, single-stranded DNA. Done. Next, we are going to move on to the RNA virus. Right? Now we have the RNA genome, double-stranded RNA. Then we move on to the single-stranded RNA. Okay, single-stranded RNA with a plus sense, then single-stranded RNA with a minus sense. Okay, first double-stranded DNA, single-stranded DNA. DNA finish. After that, we move on to double-stranded RNA, single-stranded RNA. Single-stranded RNA, that is positive, plus, and negative, minus. Okay. After that, the last 2 classifications, these are a combination of RNA and DNA both. Okay. So they have single-stranded RNA genome that is replicated with DNA as well. That contains RNA plus DNA. And that is single-stranded, which is class 6. And class 7 is double-stranded RNA that replicates with the RNA intermediate. Okay, so single-stranded RNA plus DNA, then we have double-stranded RNA plus RNA intermediate. Okay, so this is how we have covered the Baltimore classification, right? You don't have to remember these viruses, not important. Just remember this. Example of animal viruses. These can be asked in MDCAT, especially hepatitis, chicken anemia, polio, influenza. These are very important. Okay. So I'm moving on to the next classification of virus on the basis of shape, right? So we already discussed some of these before. The first one is helical virus. The example which we studied is the TMV, tobacco mosaic virus, right? And it infects plants, also rabies in animals. Okay? So what does it have? It has got a spiral shaped RNA that is enclosed with a spiral shaped helix, that is the capsomere, that is basically made of capsomeres, and that is the capsid. Okay, the capsid twists around and it encloses its genetic material. Okay, this is the genetic material, this RNA, which is present inside the capsid, and this capsid is made up of capsomeres arranged in a cylinder like shape. The next one we have is the polyhedral virus. Okay, first, we had the isosahedral virus that contains 20 faces and 12 vertices. These are the polyhedral virus. Now, what a polyhedral virus is made up of? Their genetic material is made up of DNA. As you can see over here, they have a covering of capsid and they have glycoproteins on their surface, right? These glycoproteins help in anchoring, right, to other host cells and other viruses in order to survive, right? And they cause large amount of illnesses from pink eye, pneumonia. They are composed of genetic material that are surrounded by capsid, and usually they have 20 triangular faces. Similar to isosahedral viruses, polyhedral viruses also have 20 triangular faces, and the examples are pneumonia and pink eye, right? Next, we move on to the spherical virus. Spherical virus, the most common example, which we already know, you have already seen the shape a lot before, and the example is the coronavirus, right? So coronavirus, the structure is that the inner genetic material is made up of RNA that is surrounded by a circular structure of capsid that is made up of capsomeres, and surrounding that they have what? They have got glycoproteins. Okay, they also have an envelope which is spiked sugary proteins that assist in sticking to and entering the host cell. So what they do is that they basically stick and they anchor to the host cells, and in order to enter inside the host cell. All right. So this is a very important coronavirus. The next we know that is complex virus. Very easy. The genetic material is made up of DNA. There is a head that is made like an isosahedron. Next we have this, the sheath that is made just like a helix, and we have the tail fiber as well, right? And these don't follow a single shape, right? These follow multiple shapes. Okay, that is, these are known as complex. And the main example is bacteriophage. You don't need to remember the life cycle of a bacteriophage, the lysogenic cycle for MDCAT. It has been excluded. Okay, you just need to do the classification of virus. You need to do HIV and AIDS, which I have made this presentation, so I'm going to teach everything to you. Okay, if anything is not clear, just write down in the comment section and I'll be glad to answer. Right, so moving on to our topic. Bacteriophage basically is bacteria and they resemble a lunar lander, and they're composed of a polyhedral head, as I explained it to you, and a body which is just like a helix, right? And next we have tail fibers. Why are the tail fibers present? So that they can attach to a cell membrane, right? Tail fibers basically help in anchoring, and they have to attach to the cell membrane of the cell so that they can easily perforate and enter inside the cell, right? So all other of them have got glycoproteins that help in anchoring, but complex viruses like bacteriophage, they have got tail fibers which help in anchoring. Okay, so I hope it's clear till here. Next, we move on to the classification on the basis of the presence or the absence of envelope, right? First, we studied, earlier we studied, I mean, that there are some viruses that have got envelope. Envelope is made up of what? It is made up of lipoprotein, right? And after that, there are some spikes on it that are made up of sugar, most probably glycoprotein, right, which help in anchoring. Right, now there are some viruses that contain this envelope, some of them don't contain this envelope. Those who don't contain this envelope are known as naked virus, and these naked viruses are resistant to harsh conditions. The examples are adenovirus, poliovirus, parvovirus, and norovirus. You need to remember the examples. That question can be like does polio virus contain an envelope or not, lipoprotein envelope or not? So you have to enter in a true or false statement, that does it actually have an envelope or not. So if you remember these examples, these are very common examples, then you'll only be able to solve this question. And the envelope viruses include lots and lots of examples. Wait, let me just clear the area. Okay, so the examples are HIV, influenza, herpes, rabies, hepatitis, and measles. Okay, so lots of common diseases are present over here, and the naked viruses are actually resistant to harsh conditions. Right, now see over here. This is the envelope, right? And these are the glycoprotein spikes, which are encoded in the host derived lipid bilayer. What happens is that these glycoproteins, they help to penetrate into the host cell membrane. Okay, why? Because we you know that we studied the fluid mosaic model, and that they had a lipid bilayer, right? Lipid bilayer. So these glycoproteins can help the cell to easily invade inside the host cell. All right, so this virus can be easily adapted to that condition, and these are non-enveloped viruses: adenovirus, enterovirus, poliovirus, right? And this is a nucleocapsid. You see that this structure, the yellow structure and the blue structure, are absent in the naked virus. It means that the lipoprotein envelope, as well as the sugar which is on that envelope, the glycoprotein, that is also absent. It only contains the nucleocapsid. Okay, and inside that, both of them have the similar structure. Both of them have either RNA or DNA, which is their genetic material, and outside nucleocapsid. So in non-enveloped viruses, the structure remains still here, whereas when the virus is enveloped, so they have a lipoprotein coat plus glycoprotein that helps to invade the lipid bilayer of the host cell. Okay. Now we have several examples of the enveloped virus, that are SARS-CoV, very important, coronavirus, MERS-CoV, right, Ebola, influenza, right, pox virus, hepatitis B, hepatitis C. These are very important, and I'm going to share these presentations with you as well. So there's a link down below in the description box. If you like this presentation, let me know. I'm going to share it with you. Based on capsid symmetry. Now there's a classification on the basis of the capsid symmetry, which we have already done. That is why I haven't explained this right here. The first one we did is isosahedral, the second is helical, and the third one is complex, right? Complex bacteriophage, it is both the head as well as the tail. That is why it is known as the complex structure. So we already discussed this. Next, we move on the basis of the site of replication, right? On the basis of site of replication and on the basis of genome, these are classified. The first one are DNA The second one are RNA, as we already studied in Baltimore classification. But the ones whose genetic material is DNA, they mostly replicate inside the nucleus. Okay, please remember that the site of replication is the nucleus, except pox virus, which replicates inside the cytoplasm. So this is a alarming point. You should remember this for the MDCAT. And the second one are RNA virus, which usually replicate in the cytoplasm, but there are some exceptions. For example, influenza and retrovirus, both of them have RNA as the genetic material, but they replicate in the nucleus and they don't replicate in the cytoplasm. So these are the exceptions, right? All right. Now we move on to the classification on the basis of the range of the host, right? The first one is animal virus, the second plant, and the third one are bacterial virus. So you don't need to remember these. I just add them because they are part of the classification. The ones which reside in animals are herpes, influenza, HIV. Most of them are animal viruses, and the viruses which are included in your MDCAT syllabus are also the RNA viruses, not sorry, these are the viruses which infect plants, not the viruses which infect. I'm so sorry. I'm saying that the viruses which are included in your MDCAT, those are the animal viruses, not the plant viruses. Okay, so you don't need to focus on plant viruses or complex virus, just focus on animal virus. So the example of plant virus is TMV and bacterial virus is T4 macrophage, right? Next, we move on to the on the basis of pathogenicity. What do you mean by pathogenicity? That is when the virus invades inside the host cell. Where does it act? Where does it go and it radiates its effects to? Where does it cause harm? Where does it cause side effects? So there are some viruses which cause harm in the respiratory system, which are influenza, common cold, adenovirus, respiratory syncytial virus, coronavirus, right? Next, we have enteric virus. What do you mean by enteric? Enteric means that they invade the gut, okay? The immunity system. These are polio, rota, noro, and hepatitis A. Next, we have neurotropic. Neurotropic means that they invade the nervous system. They they just attack on the nervous system. These are rabies, polio, and the JC virus. Next, oncogenic virus. Oncogenic means it means that they produce tumors, production of tumors, which is Epstein-Barr virus that causes Burkitt's lymphoma. And this is slightly higher than your level. It's better that you remember them, but if you even don't remember them, no issue. HPV, hepatitis B and C, which causes hepatocellular carcinoma. What do you mean by carcinoma? Carcinoma simply means cancer. So don't get confused in all of these. These are the viruses which cause cancers, right? For example, the question can be that what does herpes HPV lead to? So you say that it can lead to cervical cancer, right? Why? Because it is an oncogenic virus and it causes tumor. So we are done with the first part of our lecture. See, we are done with this 1.1. Classify viruses on the basis of structure. We did it. Number of strands. We did it. Diseases. We did it. And on the number of hosts, which are animal, plants, or they are complex. Right? So we already did all of that. Now we move on to the second part, 1.2, which is the AIDS and HIV infection. Now let's read the learning objective. Identify the symptoms, mode of transmission, and the cause of viral disease, which is AIDS. You just have to do the symptoms, mode of transmission, and right? But for your clarity in concept, I have made a few slides on the structure of the HIV virus, right? So let us start from the HIV virus. Okay. All right. Human immunodeficiency virus. It is from the Baltimore group 6, which is a small stranded RNA virus. So what you see over here that the genetic material is RNA. It's not DNA, it's RNA. Please remember this. Okay. And after that, what do you have? After that, see, you have got the nucleocapsid protein 24, glycoprotein GP120, and glycoprotein 41. You need to remember these. These are very important. Okay, and the matrix protein, the name of the matrix protein is P17. And there's a reverse transcriptase enzyme as well for the conversion of RNA to DNA and vice versa. Okay. Now we move on to the HIV virus. HIV virus is a retrovirus. You need to remember this. It belongs to the family Retroviridae. It causes AIDS. Why does it cause AIDS? AIDS because it destroys the CD4 T lymphocytes. Lymphocytes are what? Lymphocytes are kind of white blood cells, right? It destroys the immune system of the body. It destroys the white blood cells, right? And after that, it causes AIDS. The genome is the positive double-stranded RNA, two copies, which means that it is diploid, and it carries the reverse transcriptase enzyme, which converts the RNA to DNA inside the host. Okay? See, as we saw over here, this is the enzyme reverse transcriptase. It helps to convert the RNA into DNA, right? Now the structure of the virion. The first part is the envelope. It is the outer covering which is derived from the host cell membrane. What does it do? It basically, steals the cell membrane of the host and it forms its own envelope. Okay, this is the envelope. The red you can see. This is the envelope or the lipid bilayer membrane which it has stolen from the host. And this contains glycoprotein GP120 and GP41. Okay. Okay, this is a surface glycoprotein. It binds to CD4 receptor and co-receptor. CD4 is what? It is a kind of white blood cell. It is kind of a leukocyte, right? So it binds to these cells and then eventually kills them. And GP4 is a transmembrane glycoprotein which mediates the fusion with the host cell membrane. Okay, it helps the fusion with the host cell membrane. Next is the matrix. It lies beneath the envelope and it is composed of P17 protein. See. This is the matrix, the blue one, this one. It is the matrix and it is made up of protein that is known as P17. Okay. What is the function? It provides structural support, helps in the assembly of virions. Okay. When a lot of virions are collected, it helps assembling them and it also provides structural support. Okay. Next, we move on to the capsid. What is a capsid? You have to answer this in the comment section. Now, what is capsid? You're going to tell me now. It is made up of P24 protein. Just remember the names of the proteins, right? And it closes the genome, right? Capsid is basically a core made up of capsomere, and inside we have got the genetic material. So the first layer, the red one, is the envelope. The second layer, the blue one, is the matrix. The third one, again the red one, it is the capsid. That is made up of capsomeres and it encloses the RNA. Okay. Next, we have the genome. It is made up of small threaded RNA. It contains 9 genes. You need to remember this, but don't go into a lot of details. I just added this information. You can read it, but I won't suggest that you spend time learning it. Okay. Next, we move on to the viral enzymes, which are inside the capsid, which is reverse transcriptase, integrase, and protease. Okay, just remember reverse transcriptase, it's very important. Now we move on to HIV, which is human immunodeficiency virus, right? And it causes AIDS. All right, now what are the symptoms? You need to remember the symptoms as a case can come like a person has got experiences weight loss, chronic diarrhea, lymph nodes, mouth problems, right? And his CD4 count is very decreased. CD4 count is very less. So what do you suspect? What is happening in this patient? So you're going to write that the patient is undergoing, and the patient has got a disease that is known as AIDS. Okay, that is due to HIV, right? Now, fever. Fever is how is the fever? It is long lasting. We also have got night sweats. Then weight loss is very severe. Chronic diarrhea. Chronic means that it lasts more than 3 months. Any symptom which lasts more than 3 months is known as chronic. It means that prolonged. Okay, it is long lasting. Next, we have swollen lymph nodes. Lymph nodes are present in the neck, armpit, groin, and these will be swollen. Next, we have mouth problems that is known as white patches in oral thrush. Oral thrush is what? Thrush is basically a fungal infection which is caused by a candida, right? So basically when a person becomes immunodeficient, right? Immune system of a person is suppressed because the white blood cells of a person are gone. Okay, the this binds to CD4 cells, right? The glycoproteins on the envelope, they bind to CD4 cells and they eventually kill them, right? So that is why what happens, every infection, every fungus which is present in the atmosphere, which does not infect a very healthy person because they have white blood cells, they can infect a person with AIDS, right? That is why it causes oral thrush that is characterized by white patches in the mouth and lots of sores. Next, we have skin problems, rashes, purple spots that are known as Kaposi's sarcoma, and frequent infections. Let me show you a picture of the oral candidiasis and Kaposi's sarcoma. Wait a second. Um, okay. See, this is Kaposi's sarcoma. It looks so painful, right? And also, I'm gonna show you oral thrush. See, white spots, sores, flaky skin. Okay, this is known as the oral thrush. And next, we move on to frequent infections, pneumonia, tuberculosis, flu-like illness, tiredness. Obviously, a person's immune system is suppressed. He's taking medications, right? He has got lots of sores in the mouth, pneumonia, tuberculosis. So he will eventually experience weakness and tiredness as well. Next, we have memory or brain problems, just like dementia. It also infects the brain. It causes lots and lots of infections. And why does it cause lots of infections? Because immune system is suppressed, so now all the foreign particles and foreign bodies which are present in the atmosphere, they can easily infect that person. Now we move on to the mode of transmission of HIV. Okay, so the first one that is sexual transmission, and it is the most common. Whenever it is asked to you that what is the most common way of transmission of HIV, so you will say that it is sexual transmission. Vaginal, anal, or oral sex with the infected partner, it can cause HIV. And if anyone has multiple partners, so that is, it can increase the risk of HIV or any other STD that can also lead to HIV. Next, blood and blood products, the ones who basically do IV drug abuse by sharing needles and sharing syringes, they can cause HIV. Contaminated blood transfusions in hospitals or in setups where there is not adequate needles or adequate equipment given for blood transfusions, their contamination can occur very easily, and risk of HIV can increase. Right? Next, we have needle stick injury. Needle stick injury in healthcare workers. What happens is that the needles which they have in, put in the patient's hands or cannulas, etc., these can infect the healthcare workers as well if they are not disposed properly and if the healthcare workers don't take care. Now, if a mother has HIV, she can also transfer HIV to her child during pregnancy. That is known as transplacenta via her placenta when the blood exchange occurs between a mother and a baby. So, HIV virus can also be transferred into that baby. Also, during delivery, when a baby is exposed to mother's blood and secretions, so HIV can be transferred, and through breastfeeding through mother's milk, HIV can also be transferred. Okay. There are other routes which are organ tissue transplantation. For example, if there's a person who has donated a kidney for organ transplantation, right? And this person has got HIV. So if any healthy person undergoes transplant and then the HIV kidney is placed inside the healthy person, so then the healthy person will also develop HIV. And artificial insemination with the infected donor semen. Okay, so when someone donates semen and it contains HIV virus, so it can also lead to HIV in a normal person if that person takes that. Okay, now we move on to the stages of HIV. The first stage, patient will not know that he has got HIV. Okay, live his life just like he was living before. Patient has HIV but no symptoms. Asymptomatic means no symptoms, and this may last for several years, like 5 to 10 years. Patient cannot know that he is HIV positive, and the virus replicates at very, very low levels. That is why the patient does not know. Why? Because the virus is not, some viruses rapidly act, while this is slowly and steadily acting. This is chill, okay? So the person is also chill. The person does not know what happened. And CD4 count gradually decreases. Why does it gradually decrease? Because the HIV, it would slowly and steadily start eating all the lymphocytes and it would slowly eliminate all of them. So this may take 5 to 10 years, right? Person may not know and there will be no symptoms. There are maybe some symptoms that there will be generalized lymphadenopathy. What is lymphadenopathy? It means lymph node will be enlarged. As we saw earlier, that there will be the swelling of the lymph nodes in greater than size and in greater than 3 months. Okay, so this will be persistent, right? So the lymph nodes will be swollen, but elsewhere, person will have no symptoms and he will not feel anything. Okay. Now we move on to the age-related complex, ARC. The first stage was asymptomatic. The second stage is age-related complex. It is early symptomatic stage, also known as pre-AIDS. First it was AIDS, now it is known as pre-AIDS, which means before, slightly before AIDS. Like it has got, symptoms, but they are mild. Unexplained weight loss greater than 10% of the body weight. For example, if a person's weight is 60, he will undergo greater than 10% of the body weight loss, that is greater than 6 kg. Okay, if a person is of 60 kg, he will undergo a weight loss which is greater than 6 kg. Okay, and chronic diarrhea which is greater than 1 month. So these symptoms are mild and they will eventually increase. Why? Because this is known as pre-AIDS. This is early symptomatic AIDS. Next, we have prolonged fever or night sweats and recurrent oral infections, which is oral candidiasis, herpes, oral hairy leukoplakia. You don't need to remember leukoplakia or others, just remember candidiasis as it is written in your Federal Board book as well as Punjab Board book. Next, we have fatigue, which means that a person will have tiredness, malaise, anorexia. Anorexia means basically uh, anorexia is like, the failure to eat, okay? That is known as anorexia, right? So a person will have a few symptoms like fever, night sweats, infections, oral infections, chronic diarrhea, and also weight loss, anorexia, and lots of fatigue. And his CD4 count is written over here. It is less than 500 cells per microliter. So when the blood culture and sensitivity test and when the FBC, the full blood count will be done. So what will happen? The decrease, that it will be decreased. Why will it be decreased? Because slowly and steadily HIV has started to go to your body and the CD4 count will be less than cells per microliter. Okay. Patient recurrent bacterial infections and minor opportunistic infections. So there will be minor infections which keep on in the person's body and the patient will have bacterial infections. Why? Because as soon as he, goes somewhere or whatever, he is exposed to the outside environment. So bacteria will invade his body and it will start to destroy it. Why? Because the CD4 count is slowly and steadily deteriorating. Okay. And in order to, I'm sorry, and in addition to this, you will also have candidiasis, right? And sores in the mouth, tiredness, weight loss, diarrhea, night sweats, okay, fever. So all of these, failure to eat, he would not like to eat food, okay. So these are the stages. The third stage, this is not pre-AIDS. Now this is the, this is proper AIDS and this is known as full-blown AIDS. It is the final stage and it is the most severe stage. CD4 count, it drops less than 200 micro per liter. You need to remember this CD4 count. In the first stage, in the asymptomatic stage, the CD4 count remains same. It slowly, very, very slowly decreases in almost 5 to 10 years, right? Next, we had the pre-AIDS stage, and in this the CD4 count was less than 500 cells per microliter. Right. Next, we have the full-blown AIDS. In full-blown AIDS, the CD4 count will be less than 200 cells per microliter. Okay. And the features are severe opportunistic infections. Severe infections, for example, fungal infections, pneumocystitis, gyrovc, pneumonia. You don't need to remember of the MBBS level, which you are gonna be in next year, inshallah ta'ala. My prayers are with you. You just need to remember that there are certain fungal infections, candidiasis, pneumonia, right? And there's one disease which you need to remember. See, Kaposi sarcoma, very important. These are, as I showed you before, these are the sores on the skin. Sores on the skin. And in parasitic, you need to remember toxoplasmosis. Bacterial, you should remember tuberculosis. Okay, these are just important. Don't focus on the rest of these. Now, AIDS defining malignancies. Due to AIDS, there are certain cancers which occur. The first one is Kaposi sarcoma, as I showed you before. Next is non-Hodgkin's lymphoma. You don't need to remember this. And cervical carcinoma, the carcinoma of the head and neck. All of these are very important. Okay, in easier terms. Neurological disease. It contains dementia, encephalopathy, and neuropathy. Just remember dementia, the other rest two are not of your level. Severe wasting syndrome. There's a wasting of muscles. A person is tired, he's fatigued, not able to walk, and he has got severe chronic diarrhea, weight loss, not able to eat, severe sores in the mouth, severe sores on genital areas, right? Kaposi sarcoma, severe sores on the skin. So, like, a person's life is destroyed. Patients are highly immunocompromised and their life expectancy is short without treatment. Okay, so there now scientists say that they have found a permanent treatment for HIV, but still it is not present over here where we live, and a patient will be highly immunocompromised and life expectancy is very short. Okay, so we are officially done with our first topic of the MDCAT biology. Okay, so if you have any questions, let me know. I'm giving a phone number in the description box below. Okay, if you want all the links to the presentations which I'm gonna teach in the next 15 to 20 days before MDCAT, it will be, a sort of a crash course. So just text that number and I'm gonna add you guys into a group, and I'm going to share all the presentations and all the notes and all updates related to MDCAT, not just biology, but all the subjects. Okay, English, logical reasoning. I've just started to make content. So I won't be able to do more than biology this year, but for the next cycle, inshallah, we're going to do everything together. Okay, so I'm ending the class over here. If you guys have questions, let me know. And see you guys in the next class, inshallah ta'ala. Right? Till then, take care and best of luck for your MDCAT. If you have any questions, kindly ask me. Thank you so much, Allah Hafiz.